Welcome back, everybody, to the Curing Retinal Blindness Foundation YouTube page, and welcome to another episode of Eyeing Progress. I'm your host, Mitchell Smedley. This is the show where we get you engaged and excited about all things going on in the field of vision research. And today we're back with another fantastic guest. So excited to talk with Dr. Irene Maumenee. Dr. Maumenee, how you doing today?
I'm doing just fine. Thanks for having me. It's a real pleasure.
Thanks for coming. Yes, it's
Um,
it's a pleasure to be speaking with someone so impactful and who's had such a storied career in the vision research field. I know that when the diagnosis came down for my older brother, you were the person that my family was referred to as kind of the one person that was really getting things going in this field. That was 24 years ago. So it's great to kind of have this full circle moment and get to explore all things vision research with you. So I guess we'll just start at the beginning. How did you get involved in this field of vision research and all things blindness? Is this where you started in your career, or was there something beforehand?
no, I started very much with inherited blindness. I was a medical student in Switzerland, and in the Swiss valleys, you have a lot of blindness. And I was there in the University of Geneva, and there was Franceschetti, who was a very famous ophthalmologist at the time, and who had one of the centers for inherited blindness. And it covers basically Switzerland and Italy. And then in the north of Europe, there were people like Francois in Ghent, in Belgium, and in the Netherlands, and in Sweden and Denmark and Finland, basically in those countries where there were islands or small communities, small rural communities, with a lot of inbreeding. Now, ophthalmologists had become aware of a high frequency of inherited blindness and wanted to solve this. So I was a medical student in Geneva, and during one lecture, a geneticist at the eye department in Geneva just gave a very eloquent discussion about genetics in ophthalmology. And decided I was going to join them, after. So I was basically, let's say, a couple of years into medical school at the time. And I said, I go back, and I went back to Germany to finish medical school. And then I returned to Switzerland, and they gave me a job to sort of write a thesis and start out my career. And it was a very exciting time and very good. But this may sound, it was a second thought, but basically, as children, we were already working on genetics. Um, so I had a cousin who was very, very much into biology and everything that was living and about. And he and my twin brother and I, we collected a collection of snail. We made a collection of small snails, and we bred them or tried to breed them on our balcony. Um, it's a, they're special shell colors that come in snails, and they're beautiful, and we never got anywhere. And it was really, snails, they're facultative hermaphrodites. They only breed when they have other snails around, but otherwise they can also be hermaphrodites. So, we had no idea of all of this. We were 6, 7, 8 years old. But I never let go of the field of genetics. I found it fascinating. And so, that's how it changed into blindness, because that's what they taught in Geneva. I wanted to go back to Geneva. I met a mentor, and so it all started from there.
That's amazing. I The snail story, everyone has that kind of moment where they feel that this is sort of my calling, and for you to have that so young and be so interested in a field like biology. I got my first taste of biology in just, a high school class, and I hated it. And I
Yeah.
just was like, how is this, ever going to be useful in any way? But then, obviously I start looking into like the vision research and all that stuff. And it's like, oh, this actually impacts my entire life. So really interesting that it kind of started so young for you. So what was kind of the catalyst, I guess, from working with, uh, those folks over in Geneva and in those communities? What brought you over? I know that you did some work in the United States over the years. What brought you over here?
Actually, a population geneticist from the University of Hawaii came to Geneva, and he looked at my data and he said, "Why don't you come and spend 3 months with me in Honolulu?" Um, and I said, I didn't want to go to Honolulu. And, who doesn't want to go to Honolulu? But I didn't want to go. And my mother said, "You're crazy. Of course you need to go." She didn't really anticipate what was going to happen because I stayed a year in Honolulu, and then on my way back to Germany, I went to Hopkins, and I worked with McKusick at the time. And then McKusick offered me to stay there longer. And, and from just coming for 3 months, I'm still here, some 50 years later. So it's
50
really,
years. Wow.
no, it's actually, sorry, it's 60 years later.
60 years. Congratulations
60
on such a
years
long
later.
career.
That's exactly right. Yeah.
Wow.
So,
Hey, we're happy that you did come over for those 3 months. And you're right. Who doesn't want to go to Honolulu? Talk about taking the scenic route.
yeah,
That
there
must
we
have
go.
been awesome. So, how did your work change when you
Uh,
came over
uh,
to the United States?
uh,
Any new projects that you picked up?
Because, what was really the fascinating part at the time was population genetics. So while I was in Honolulu, there was not even an institute of neurologic disease, of blindness. The NEI didn't exist. It was part of neurologic disease and blindness. They were fused. And a neurologist came through Honolulu, and he had heard about an island in the Pacific where a lot of people were blind. And my boss, Newton Morton, he came to me, and it was Dr. Brody at the time, and said, "Do you want to go to the Pacific?" And I was already in Honolulu. I said, "I'm sure I go to some small islands in the Pacific. I go a little further." And so I started a study on inherited blindness on Pingelap. And you may have heard about Pingelap eye disease in your time, or if not, then read the book. It's really fascinating. Oliver Sacks wrote about that island, but we started the study there and ultimately found the gene. So it was really doing population genetics and then molecular genetics was a fascinating experience. And while I was in Switzerland, it was still all descriptive, and the basic work and research work that has happened over the last 60 decades wasn't even dreamt of. So it has really made there's been a tremendous progress in the field of inherited blindness, from description to population diversity to DNA analysis to gene therapy. It's extraordinary what has happened. And none of us thought that far ahead.
Yeah, you're absolutely right. That was my next question. Sort of, the current state of progress. Sometimes I know for patients, for families, it can feel like, the wheels are turning so slowly. But would you say that the rate of progress we're seeing now in genetic diseases and stuff like that, has that actually picked up in speed compared to what you expected when you first got into this, or in the first couple decades
I
of
never
your
thought
career?
it would go that far in my lifetime. So this is far ahead of what I anticipated. And it's really the Human Genome Project which has pulled it off. It's the people who um, thought it through and fought for it and got it passed by the government to put the money into it, which then made the difference. And it was a hard, it was a visionary approach, and it was a lot of work that went into it. So I think it's fantastic.
Yeah, can you, for those that might not know, kind of describe what the Human Genome Project is and then its effect on inherited blindness research?
Sure. The Human Genome Project, it started in the late 1990s, and people had been thinking about it for quite a while. So people had created what's called OMIM, Online Mendelian Inheritance in Men. McKusick had done that at Hopkins, where all the patient diseases were cataloged. And now what was necessary to link that catalog with diseases to the DNA analysis. So DNA analysis had become possible, but you had to also do it at a big scale. And so people went out to sequence DNA from some 25 people initially and try to sequence it all, going from, chromosome 1 to the X and Y chromosomes. So doing all the chromosomes, there were tremendous gaps because some areas were easier to sequence than others. So it was very incomplete. When they first declared it was complete, and that was under Clinton, I think I hope I have the year right, in 2003, but somebody may want to check that. So it was declared being complete. But the last complete version came out maybe a year ago. So there have been many complete claims to completeness. So it has been going forward, and there's still holes in there. But it's really already at a level that's very satisfying to have basically everybody's, to have normal DNA, whatever normal is, sort of a range of variation at every point in the human genome. The human genome has 3.6 billion base pairs, and their variation just about at every one of these pieces of DNA. And so they're quite well described in tremendous databases now. So you can really assess whether a change that you find in a person is significant or not, in which gene is it, the for CRM1 is on chromosome 8, and you have all the the variations, a lot of variations are known. And so um, people have, basically information that will be relevant to all the patients, or to most of the patients today. It's still not complete. Not every patient has been correlated, or every disease has been correlated with a specific gene and specific mutations. So this work will keep going on for the next 10, 20 years to
Sure.
bring it to a level that is really, really useful for everybody.
That's awesome. So, do you think that the mapping of the human genome is what allowed these technologies like gene therapy and gene editing to really take off? Is that kind of the baseline for all of these things?
Yes, you had to know what you were dealing with and how you can just basically cut out a piece of DNA and replace it with a healthy part. How you can even put it, change patients, or the work in the dogs. That was then a first step to move forward and do it in humans. All these things were based on the ability to sequence a human genome and to replace parts, to compare it, to link it to diseases. So these all took decades to get it to a level where it is now. A good 30 decades, so 3 decades, 30 years.
Wow, that's really impressive. So
Yeah.
it seems like the completion of that in 2003 and throughout the last 2 decades. I know that when my family was referred to you and the doctors told us, before they told us to talk to you, and you actually knew what was going on, and you've been incredible in pushing all of this forward. They told us that there was, no hope, that maybe in 50 years. And now I think,
Yeah.
you said
Yeah.
it yourself that seems to be accelerated now, and it's beyond anything we could have hoped for.
Sure. And but there are still like concerns. There's some genes and some mutations that lead to total absence of the eye, for example. You're not going to manipulate an eye and DNA to give a person who has no eyes from birth normal-sighted seeing eyes. So there's the severe end that really is difficult to handle. But then there's also the milder end, and you have possibilities of interfering. Um, so there has to be caution in being optimistic for everybody. There is a limit what people can do certainly at this point.
Sure. So kind of moving from the past sort of to looking at it from now into the future. What do you think sort of the biggest hurdles still to go in this research journey will be, some of the biggest obstacles before we find ways to cure a lot of these inherited diseases?
I think one big problem is that you use a virus to to put in normal DNA to enter the cell and enter the cell and produce a normal protein, which that's what DNA does. It goes from DNA to RNA to the protein, but you have to get the normal protein into the cell. But what's happening is that it's not clear that whatever you do in the way of gene transfer is going to last a lifetime. So you have to come up with methods that are long lasting. And that really hasn't been fully achieved. That is one thing, and I think a very critical one. Not that you cure somebody and then you go back, and 10 years later, they're losing their vision again, which is a concept that nobody wants to deal with. And that's just very difficult. But then what also happens that genes are big, and everybody has their own mutation. Are you going to create gene therapy for everybody individually? It becomes just so costly that it really can't be done. So you have to come up with methods that are kind of more neutral and that you can apply to all kinds of patients at the same time. So there are still a lot of hurdles out there to to make it work for everybody. But I feel kind optimistic like everybody in the way that people are thinking big and are working at it, and everybody sees the reasons to move ahead and get a lot done to move things forward.
That's interesting. I've had a couple guests on already, and no one's mentioned it, sort of in the gene therapy realm, that maybe it is a short-term fix rather than a long-term one, that so it is possible for, to put the protein into the cell, and then after, like you said, maybe 10 years, whatever the time frame is, the cell stops producing that protein, and you revert back to the original problem. That's really interesting.
But that's obviously, not very encouraging. So people, you don't want that. You organize a life then around sight, and you just lose all those years of learning where you should be learning how to live with your blindness and move forward and then lose it again. That's not what anybody wants to do. And so there are still hurdles there, and that are pretty significant.
Gotcha. Kind of on the contrary, where do you think, on a more positive note, that we can make the most progress in the coming years in this whole field? What do you think the kind of bright spots are?
What are the bright spots? I think, you know, um, people are learning. Well, I think they're first of all, they have to complete the that every disease that you have, you know the gene and you know the mutations. I think a lot of progress at present ought to have happen in giving every person a correct diagnosis fast so that they can know what's going on. And that happens more and more, so that patients who come to a physician's office are told, what's your disease. They'll do whole genome sequencing and figure out exactly what's wrong with your eyes. Once
Yeah,
you
so
know
you're not having those
exactly what's wrong.
misdiagnoses. Yep.
And before, we had no means of doing that. So that's happening now in a very positive way, so that people get a mutation-specific diagnosis so that they can look forward and, and figure out what is my personal prognosis, what is my situation. A lot of the information is in the public domain, and people can go and read up of where things stand, learn about it, learn about their own disease. Whereas before, physicians were more sacred, were not approachable. But I think in general, physicians now try to give the information to the patients and create a collaboration between patients and physicians to move these issues forward.
Gotcha. Sort of in the same vein, what do you think is the next big breakthrough, or the thing you're most excited for coming down the pike in vision research?
Well, we are going to our research meeting. I leave on Friday. It's the annual research meeting called AVO that's going to take place in Seattle. And we will
Oh,
hear
okay.
what people are doing and what is the latest. I think what's really happening is you get more and more understanding about the human genome and how genes interact. Um, I think the All of Us program is very exciting, which is a program which tries to sequence first 1 million people in the US, and they would like to go beyond it and sequence more and more people so that people can become aware before they have offspring of what their risks are. Or that they understand their own disease better. I think that's a tremendous progress. Um, what else is going on? I think pharmaceutical industry is very much behind it. So they really want to bring the disease to treatment because, well, they see their mission in it. And that's happening more and more. Um, and if you look at it, it's that the insurance companies, they are going for it too. They are paying for it so that it's not just asset dependent whether your disease can be treated. Um, there is Duchenne muscular dystrophy, which is a fatal muscular dystrophy that now is coming to treatment. There is sickle cell disease, really pretty traumatic, again, fatal, very compromising, life compromising, and quality of life changing disease that's coming to treatment. And they all cost, let's say, 2 million per person to treat,
Wow.
which
Yeah.
seems horrendous values. But the numbers are so small that the insurance companies and the government is paying for it. So it's
Interesting.
happening. It's happening. It's happening. And
That's really good
before,
to hear.
yeah, and before, when the API 65 treatment came in at, let's say, close to 1 million per eye, people were very upset. They thought it was really outrageous. And but all the other diseases come in that same ballpark. And people have gotten accepted to these high numbers and they are taking it, it's happening.
Really interesting. You're the first person to kind of ballpark what the treatments sort of cost, and it definitely sounds like the pharmaceutical industry and the insurance industry. There's profit to be made here. So yeah, it stands to reason they're behind it. But also, like you said, seeing the mission and seeing that this is the cutting edge of research. So before we go, any final thoughts? Anything we didn't get to that you're excited about or you wanted to share with us?
I think you bring me on again, and I talk to you again after the research meeting at ARVO, because let's see
Absolutely.
what new things are coming out during that meeting and what we hear. Because it's basically all about genetics, or a lot is about genetics during that meeting. And so we'll hear, what are other approaches that are ready for the big opening, so in a week I will know a lot more.
Awesome. Well, we'll have to do a Dr. Maumenee eyeing progress part two.
There we go.
Thank you so much for joining us. Have a great time in Seattle, and yeah, we'll be sure to check back in with you and see what you find out. But really appreciate your time. Really appreciate. I mean, like you said, those 60 years of your dedication to this mission. We cannot thank you enough. And best of luck to all that you continue to do in this field and in your entire life. And thank you so much. So that's going to do it for this week on eyeing progress. But we'll be back soon, maybe with Dr. Maumenee once again, and maybe with someone else as we continue to get everyone engaged and excited for all things going on in the exciting field of genetic vision research. Thank you so much once again, Dr. Maumenee, and we'll see you next time here on the Curing Retinal Blindness Foundation YouTube page.
You're welcome. It's my pleasure. Thanks for asking me.